before you noticed. You're not the majority organism in your own body, not even close. The human body contains roughly 30 trillion human cells. It also contains, by current estimates, somewhere in the range of 38 trillion microbial cells, bacteria, archaea, fungi, and other organisms that live on and in you, that have always lived on and in you, that were there before you had language for them, and will be there in some form until the whole system goes. The number has been revised downward from earlier estimates that put microbial cells at 10 times the human count, but even at rough parity, the implication is the same. You are, by cell count, approximately half not you. This is before the viruses. The viruses are a separate accounting problem. The body is running right now, not in the motivational sense, in the literal, continuous, no-pause possible sense of a system that cannot afford to stop and has not stopped since approximately the 3rd week of embryonic development when the heart first went. Everything happening in you at this moment, the sodium-potassium pumps cycling in your neurons, the immunoglobulins patrolling your bloodstream, the mitochondria in your liver cells turning glucose into ATP through a chain of reactions that would take several pages to write out fully. None of it was switched on when you woke up. None of it switches off when you sleep. The biological activity that constitutes a living human body is not something the body does when prompted. It is what the body is, continuously, without announcement. The strange part is not that this is happening. The strange part is that it is almost entirely invisible to the person it is happening inside. Consider what you actually have access to, as the nominal owner of this system. You can feel hunger, which is a signal the gut sends after a cascade of hormonal changes involving ghrelin and leptin and blood glucose levels that have been running for hours before the sensation arrives. You can feel pain, which is the end product of a signaling chain that starts at nociceptors and travels through the spinal cord, and is modulated by endorphins and processed by multiple brain regions before you have the experience of something hurting. You can feel tired. You can feel warm. You can feel, occasionally, your own heartbeat. That is roughly the readout. A handful of summary signals from a system of staggering complexity, simplified to the point where they can be acted on. The rest, the several hundred distinct cell types doing their specific jobs, the roughly 20,000 protein-coding genes being expressed in different combinations in different tissues, the immune system's continuous low-level surveillance of every surface and fluid in the body, runs without a dashboard, no readout, no notification, just the ongoing fact of it. You are, in a very precise sense, mostly unaware of what you are. The microbiome is where this gets hard to hold as a single thought. The gut microbiome alone, the community of organisms living in the gastrointestinal tract, predominantly in the large intestine, contains somewhere between 500 and 1,000 distinct species in a typical adult, varying by individual, varying by diet, varying over time. The collective genome of the gut microbiome is estimated to contain roughly 150 times more genes than the human genome. These organisms are doing things. They are metabolizing compounds the human digestive system cannot process alone. They are producing short-chain fatty acids that the cells lining the gut use as fuel. They are synthesizing vitamins, including B12 and K2. They are interacting with the enteric nervous system, the 500 million or so neurons embedded in the gut wall, in ways that influence gut motility, pain sensitivity, and through pathways that are still being mapped, mood and cognition. The gut microbiome is not a passenger, it is infrastructure, and the person whose gut it is has essentially no conscious knowledge of what species are in there, what they are doing on a given day, or what the consequences of any particular change would be. The system runs. The readout does not include it. Before all of this was known, before germ theory, before microbiology, before the sequencing technologies that made the modern microbiome literature possible, the body was understood as a self-contained thing, bounded. The skin was the edge. What was inside was you. What was outside was not. That model worked well enough for most purposes. It still works for most purposes. The intuition of a bounded self with an inside and an outside is not wrong in any practical sense, but it is, at the biological level, a simplification that the last several decades of research have made increasingly hard to maintain without qualification. The edge of the body is not where it looks like it is. The self that is doing the running is doing it in collaboration with organisms that are not self, using processes that predate the species in a system that is more complicated and more shared than any intuition about individual bodies tends to include. The running was happening before you noticed. It is happening now. It will still be happening after you fall asleep, after attention has gone entirely, after the last thought of the night has gone wherever thoughts go, when the brain shifts its mode. The body does not require the awareness. It has never required the awareness. The awareness is, in a sense, the optional part. The bacteria outnumber you. Start at the lumen, the inside of the large intestine, a warm anaerobic space. Oxygen is nearly absent here, roughly 1.5 meters long, with a surface area that, if unfolded and laid flat, would cover something close to a square meter. The walls are lined with mucus, not a thin film, a layered structure, an inner layer, dense enough that bacteria cannot penetrate it and reach the epithelial cells beneath, and an outer layer, looser, where certain bacterial species live and feed. The mucus is colonized, has been since the first days of life, will be until the system goes. In the outer mucus layer and the lumen itself, the bacteria are doing what bacteria do, which is everything continuously, in a space that has been their habitat for longer than the person housing them has been alive. Bacteroides fragilis is breaking down polysaccharides, complex carbohydrates, that the human digestive enzymes upstream have left intact. Faecalibacterium prausnitzi, one of the most abundant species in a healthy gut, is producing butyrate, a short-chain fatty acid that serves as the primary energy source for the colonocytes, the cells lining the colon. The colonocytes, in other words, are running partly on bacterial waste products. The lining of your colon is being fed by organisms that are not you. The thread is this. Where does the body end and the microbiome begin? Follow it. The colonocytes are human cells. They have human DNA. They are expressing human genes, producing human proteins, doing human cellular things. They are also dependent, for a significant portion of their energy supply, on short-chain fatty acids produced by bacterial fermentation. Remove the bacteria and the colonocytes do not function the same way. Studies in germ-free animals, raised in completely sterile conditions with no gut microbiome, show underdeveloped gut linings, altered immune systems, and a range of physiological differences that suggest the microbiome is not optional infrastructure. It is part of the system. The human immune system developed, in evolutionary terms, in the presence of microorganisms. The gut-associated lymphoid tissue, the collection of immune structures in the gut wall, is shaped by microbial presence. In germ-free animals, it is underdeveloped. The immune system learned partly from the bacteria. It calibrated its responses, developed its tolerance for self and non-threatening non-self in an environment that always included the microbiome. The immune system is not separate from the microbiome. It grew up with it. The skin is the other place to follow the thread. Roughly 2 square meters of surface, and on that surface, another community. The skin microbiome varies dramatically by location. The oily regions of the face and scalp host different species than the dry skin of the forearm, which host different species than the moist folds of the elbow. Staphylococcus epidermidis is one of the most common residents of human skin and is, under normal conditions, entirely benign. More than benign. It produces peptides that inhibit the growth of more harmful species, including Staphylococcus aureus. One skin bacterium is doing antimicrobial work on your skin, continuously, without requiring any instruction from you. There is a species called Cutibacterium acnes that lives in the follicles and sebaceous glands, feeding on the sebum the glands produce. It has been living there since adolescence. It is one of the organisms associated with acne when conditions change and populations shift, and it is also, in a balanced state, a normal resident of nearly every adult human face. The same organism, depending on context, is either part of the system or a problem the system is managing. The thread continues and gets thinner. There is no clean version of the body that exists without these organisms. The clean version has never existed. From the first moments after birth, when the infant passes through the birth canal and is colonized by maternal microbiota, or is born by caesarean and is colonized differently, more slowly from the environment, from that first contact, the body and its microbiome are building the same system together. Here is the sentence that changes its mind. The microbiome is not something you have exactly, and not something you are. And the longer you sit with that, the less certain the boundary becomes. The numbers alone do not settle it. The functions alone do not settle it. What settles it partly is the evolutionary record, which shows that the relationship between humans and gut bacteria is ancient, that the core of the human gut microbiome shares deep ancestry with the gut communities of other primates, that this is not a recent accident of civilization, but a long coevolution between a host lineage and a microbial community that has been running for millions of years. They adapted to us. We adapted to them. The adaptation went both ways, over a time scale that is difficult to think about from inside a single human lifetime. The thread ends here, not with a boundary, but with a question about whether boundary is the right concept. Pause here for a moment. If you grew up being told the body was yours and yours alone, a contained, bounded self running on its own systems, this is probably the chapter where that starts to feel slightly off. Not wrong, just incomplete. What we want to know is this. When did you first realize the body was more complicated than you were taught? Was it this kind of thing? The microbiome, the non-human residents, or something else entirely? Leave it in the comments. Not because it changes anything scientifically, just because this is the kind of thing that is interesting to think about at this hour, and apparently other people are also here, thinking about it, which is one of the stranger facts about putting something like this into the world. What a cell does on a Tuesday. The ATP is already being made, not starting to be made, not about to be made continuously in a process that has been running since the cell was new and will run until the cell is not. A process that does not have a beginning within any given moment, because within any given moment it is simply ongoing, the way a river is ongoing when you arrive at its bank. Not starting, not ending, just there, doing what it does with no particular interest in being observed. A typical human cell contains somewhere between 1,000 and 2,000 mitochondria, depending on the cell type and its energy demands. A liver cell tends toward the higher end. A red blood cell has none. A neuron in an energy-hungry region of the brain may have more than 2,000, clustered toward the synapses where the metabolic demand is highest. Each mitochondrion is running the electron transport chain, a sequence of protein complexes embedded in the inner mitochondrial membrane, passing electrons down a series of carriers in a process that pumps protons across the membrane and then uses the proton gradient, the difference in concentration on either side, to drive the synthesis of ATP from ADP and phosphate through an enzyme called ATP synthase that physically rotates as it works, one of the few molecular machines in biology that uses rotation as its mechanism. The ATP leaves the mitochondria and goes where it is needed. In a cell doing ordinary work, maintaining its membrane potential, synthesizing proteins, managing its internal environment, the demand is constant and the supply is constant, and the two are kept in a balance that the cell is always actively managing. The sodium-potassium pump in the cell membrane uses ATP to move sodium ions out of the cell and potassium ions in, maintaining the concentration gradients that the cell needs for signaling and for keeping itself from swelling with water. It runs continuously. In a neuron, the sodium-potassium pump accounts for roughly 70% of the cell's total energy budget. Most of what a neuron's mitochondria are doing most of the time is keeping that pump going. The pump is not doing anything dramatic. It is doing maintenance. It is the biological equivalent of keeping the lights on, and the temperature controlled and the structure intact. Work that is necessary and continuous, and produces no particular event to notice. The nucleus is doing something different and slower. At any given moment, some portion of the cell's DNA is being transcribed, read by RNA polymerase, which moves along the double helix and synthesizes a complementary RNA strand that will be processed and eventually translated into protein. The transcription of a single gene can take anywhere from seconds to minutes. The subsequent processing, splicing out the introns, adding a cap and a poly-A tail, exporting the mRNA through the nuclear pore complex, takes longer. Translation at the ribosome, where the mRNA is read and the amino acid chain is assembled, takes longer still, with rates of roughly 5 to 20 amino acids per second, depending on the ribosome and the context. A protein of 500 amino acids takes somewhere between 25 and 100 seconds to translate. The cell is doing this for thousands of different proteins, at different rates, in different quantities, regulated by a system of transcription factors and signaling cascades and chromatin modifications that determines which genes are accessible at any given time and which are not. The cell is not expressing all 20,000 protein-coding genes simultaneously. It is expressing a specific subset, appropriate to its type and its current state, adjusted continuously in response to signals arriving at the membrane from the surrounding tissue and from the bloodstream. The membrane itself, a phospholipid bilayer, roughly 7 to 10 nanometers thick, thin enough that a single protein can span it multiple times, studded with thousands of proteins, each one doing something specific. Receptor proteins waiting for signaling molecules, channel proteins allowing specific ions to pass, transporter proteins moving glucose and amino acids in against their concentration gradients using ATP to do it, adhesion proteins holding the cell to its neighbors. The membrane is not a wall. It is an active interface, constantly in motion. Individual phospholipids diffusing laterally through the bilayer at rates of several micrometers per second, and the proteins embedded in it are not static either. Receptors cluster and disperse, channels open and close. The whole surface is a dynamic field of ongoing activity that the cell is managing and that contributes to what the cell is. Somewhere in the cytoplasm, a misfolded protein is being found. The chaperone proteins, HSP70, HSP90 and others, circulate through the cell and interact with proteins that have not folded correctly, giving them another chance to achieve their functional conformation. If refolding fails, the protein is tagged with ubiquitin, a small protein that acts as a signal for the proteasome, a large multi-subunit complex that unfolds the tagged protein and degrades it into peptides. The cell is continuously editing its own protein inventory, finding errors, correcting or removing them, maintaining the quality of its functional molecules. This is happening in every cell in the body, right now. Ubiquitin is being attached to misfolded proteins in your liver and your kidneys and the cells lining your gut and the neurons in your visual cortex. The proteasome is degrading tagged proteins and releasing the amino acids back into the cytoplasmic pool. The cell is doing quality control on itself, continuously, without pause, in a process that has been running since the cell was young and will run at diminishing efficiency as the cell ages. The cell does not know it is a Tuesday. It does not know it is any day. It is doing what it does, which is everything, all at once, in the dark. The immune system has a memory. You don't. From far away, immunity looks like protection. The immune system keeps things out, or kills things that get in, or both. That is the simplified version, the version that gets taught early and retained because it is useful and mostly accurate at the level of everyday life. You get sick. The immune system responds, you recover. The immune system remembers. Next time, the response is faster. That is not wrong. It is also missing most of what is actually happening. Closer in, the first thing that changes is the scale. The immune system is not a separate organ or a unified structure. It is a distributed network of cells and proteins and signaling molecules that exist throughout the body, in the blood, in the lymph nodes, in the spleen, in the gut-associated lymphoid tissue, in the skin, in the liver, in the brain's own resident immune population, the microglia. Anywhere the body has tissue, there is immune activity, not necessarily active defense, surveillance, continuous low-level monitoring of everything in the local environment. The innate immune system, the older, less specific branch, is doing this all the time. Macrophages, neutrophils, dendritic cells, patrolling, sampling, recognizing broad patterns associated with microorganisms or cellular damage, responding to what they find. The response is fast and non-specific. An infection triggers local inflammation within minutes. Blood vessels dilate, immune cells are recruited, the area becomes warm and swollen. This is not a malfunction. This is the innate immune system working. The adaptive immune system is the part with memory, and memory is the word this chapter wants to stay close to without quite landing on. B cells and T cells both start in the bone marrow. T cells mature in the thymus. Both undergo a process of selection, a sorting mechanism in which cells that would react against the body's own tissues are deleted, and cells that respond appropriately to foreign targets are retained. What comes out of this process is a repertoire, a vast collection of cells with different receptor configurations, each capable of recognizing a specific molecular shape, waiting. Memory B cells and memory T cells, generated after an immune response resolves, persist long after the infection that made them is gone. Some memory T cells in elderly people have been shown to persist for decades, their receptors still configured for an antigen encountered 60 years prior. The cell has been waiting, in a quiet maintenance state, for a re-exposure that may never come. The immune system made a record of something that happened before you had a conscious memory of it. The record is still there. The orbit of this chapter keeps returning to what memory means in this context, because it is not quite like any other kind of memory. Immunological memory is not stored as information in the usual sense. There is no archive, no library, no retrieval system in the cognitive sense. The memory is structural. It exists in the form of the cells themselves, their receptor configurations, their increased number relative to naive cells with the same specificity. To remember an antigen immunologically is to have more cells capable of responding to it, and to have cells that respond faster and more vigorously because they have been through the activation process before. The memory is in the population. It is a biological residue of an encounter. The encounter may have been trivial, a cold virus you do not remember having, a childhood vaccination, an environmental antigen absorbed through the gut or the skin years ago. The immune system processed it, mounted a response, retained cells from that response, and filed them in the peripheral lymphoid tissue and their bone marrow, where they are still sitting, still circulating in low numbers, still patrolling. There is a layer of this that rarely gets discussed in general accounts of immunity, the tolerance mechanisms. The immune system does not only learn to recognize foreign things, it learns continuously to not attack the self. Regulatory T cells, TREGs, are a population of T cells whose function is suppressive. They dampen immune responses, prevent overactivation, maintain the tolerance of the immune system to the body's own antigens. They are present in every tissue. They are managing at any given moment the balance between reactivity and restraint. When TREG function fails, or is insufficient or is overwhelmed, the result is autoimmunity, the immune system attacking the body's own tissues. Rheumatoid arthritis, type 1 diabetes, multiple sclerosis, lupus. Diseases with different presentations and different target tissues that share a common upstream feature. The immune system's memory and recognition system has turned in some way on the self. The same memory that protects turned sideways becomes the mechanism of destruction. The archive is the problem. The orbit comes around one last time and lands, softly, on the fact that keeps not quite resolving. You have immunological memories of things you have no other memory of. Encounters recorded in the configuration of cells that have been quietly maintaining themselves for years, that would respond if the same antigen appeared again, that carry the biological record of an immune event you cannot consciously access. The immune system's past and your past are not the same past. They overlap, but the immune system's is longer, more detailed in certain directions, and entirely inaccessible to you except through its effects. The memory is in there. You are not the one who has it. Entropy is running in there too. Something is about to be paid for. Every moment of biological order, every correctly folded protein, every maintained membrane gradient, every organized chromosome, every cell sitting in its appropriate tissue doing its appropriate thing, costs energy to maintain, and the cost is continuous. And without continuous payment the order goes, the way all order goes when energy is no longer available to maintain it. The second law of thermodynamics does not pause for biological systems. It applies to them in the same way it applies to everything else. The difference is that living systems have, for the duration of their existence, a mechanism for paying. The thread is thin and goes all the way through. Life is the temporary delay of an outcome that thermodynamics requires. The cost is visible everywhere in the cell, if you know to look for the payment rather than the order. The sodium-potassium pump, which a previous chapter described as running continuously to maintain the neuron's membrane potential, is paying entropy's bill in a specific currency. The cell uses ATP to pump ions against their concentration gradients. Left alone, ions would diffuse down their gradients and the membrane potential would collapse. The gradient is a form of order. Maintaining it requires continuous expenditure. Stop the pump and the gradient goes, not dramatically, but steadily, the way a sandcastle goes with the tide, not destroyed, just redistributed. The difference between inside and outside smoothing out toward equilibrium. Equilibrium, in biology, is death. Not because death causes equilibrium, because equilibrium causes death, or because death is what happens when the payments stop and equilibrium is allowed to proceed. A living cell is a system operating far from thermodynamic equilibrium, continuously spending energy to maintain the state it is in, and the maintenance never ends and cannot end while the cell is alive. The narrator is uncertain about how to say the next part, because it is the part that sits strangely. Entropy is not a force. It is not a pressure in the physical sense. It is a statistical tendency. The tendency of systems to move toward the configurations that are most probable, which are, for complex ordered systems, the disordered configurations because there are so many more of them. The unfolded protein is more probable than the folded one because there are vastly more ways for the amino acid chain to be disordered than correctly folded. The mixed solution is more probable than the separated one. The warm uniform room is more probable than the hot side and cold side. Living systems maintain their improbable configurations by continuously importing energy and exporting entropy, taking in ordered energy in the form of food or sunlight, using it to maintain biological order, and releasing heat and waste products that represent the entropy that has been passed on rather than allowed to accumulate. The organism is not defeating thermodynamics. It is participating in it, in a way that keeps the entropy export moving outward, while the internal order is temporarily maintained. The thread is still the same thread. The maintenance requires the payment. The payment requires the intake. The intake requires the environment to have order to give. The environment's order traces back eventually to the sun, and the sun's order traces back to the low entropy state of the early universe. And the question of why the early universe was low entropy is the question that never quite closes. The body is paying the bill right now. Your resting metabolic rate, the energy you expend while doing nothing, just maintaining the state you're in, is somewhere between 1,300 and 1,700 kilocalories per day for most adults, varying with body size and composition. This is the cost of maintenance, heartbeat, breathing, body temperature, brain activity, cell membrane integrity, protein synthesis and degradation, ion pumping, all of it running at whatever rate keeps the body in the state it is in rather than the state thermodynamics would prefer. The energy comes from food. The food came from plants, or from animals that ate plants, or from further up a food chain that traces back to photosynthesis, which traces back to sunlight, which is a star spending its own thermodynamic inheritance. The bill goes back a long way. What sits in the body right now is the end of a very long chain of energy transfer, most of which happened before you existed, and what the body does with its portion is spend it on continuing to exist, which is also what every other living thing that has ever existed has done with its portion. The improbability of the arrangement, the cost of maintaining it, the fact that it has been maintained continuously since before the body knew it was maintaining anything. The thread ends where it started, which is at the fact that something is being paid for right now in your cells at this moment, and the payment is what living is, and it has been going on without interruption, and the interruption, when it comes, will be the end of the paying and the beginning of the equilibrium, and between now and then the payments continue quietly, in the dark, in every cell, all at once. The mitochondria situation. Something happened 2 billion years ago that has not been undone. The evidence is still inside you, inside every cell in your body that has a nucleus, in the form of small membrane-bound structures with their own DNA, their own ribosomes, their own method of replication that is independent of the cell's division cycle. They divide when they need to. They have their own genome. It is small now, just 37 genes in human mitochondrial DNA, but it is there, and its existence is the signature of what the mitochondria are or were or might still be. They were bacteria. The theory is called endosymbiosis, and it is not a fringe idea. It is the mainstream, textbook account of mitochondrial origin, supported by multiple lines of evidence that have accumulated since Lynn Margulis first proposed it in 1967. The mitochondrial genome closely resembles the genome of alpha proteobacteria, specifically a group that includes the rickettsiales. The inner mitochondrial membrane, where the electron transport chain runs, is structurally similar to bacterial membranes. Mitochondrial ribosomes are bacterial ribosomes. The cell's ribosomes are eukaryotic. They are different machines doing the same job. Somewhere around 2 billion years ago, an archaeal cell engulfed a bacterial cell and did not digest it. Or the bacterial cell entered some other way. The mechanism is not precisely known. What is known is that the relationship persisted, that the two organisms began to benefit from proximity. that over evolutionary time genes from the protomitochondrial genome migrated to the host cell's nucleus, that the relationship became obligate on both sides, that neither partner can now exist without the other. The mitochondria that sit in your cells today are the descendants of that event. Descendants, not remnants. They have been reproducing continuously since the original incorporation, dividing within cells, being distributed to daughter cells during cell division, maintaining their own DNA through their own replication machinery. They have been inside the eukaryotic lineage for 2 billion years of continuous reproduction. They have changed substantially. Most of their original genome has gone, transferred to the nuclear genome or lost, but they have been there the whole time as a separate replicating entity inside a larger replicating entity. And the two have become so intertwined that the question of where one ends and the other begins is not answerable in any clean biological sense. Your mitochondrial DNA is inherited maternally, almost exclusively. Sperm cells have mitochondria, but they are typically destroyed after fertilization in a process involving mitophagy, the selective degradation of paternal mitochondria by the egg cell's own machinery. The reason for this is still debated. One hypothesis involves preventing the mixing of two different mitochondrial genomes within a single cell, which can cause functional problems. Another involves conflict between different mitochondrial lineages competing for transmission. Whatever the reason, the mitochondria in every cell of your body trace back, in an unbroken line of maternal descent, to one woman's mitochondria, and further back through her maternal line, and further back through every maternal lineage that connects, eventually, to the original incorporation event 2 billion years ago. What they are now is harder to say than what they were. They are not free-living bacteria. They cannot survive outside a host cell. Their genome is too reduced, their metabolism too dependent on proteins encoded in the nuclear genome and imported. They are, in the technical sense, obligate endosymbionts, organisms that live inside another organism and cannot exist independently. But they still replicate their own DNA. They still have their own ribosomes. They still divide on their own schedule, responding to cellular signals but retaining their own internal regulation. In cells undergoing stress, mitochondria communicate with each other through a process involving the fusion and fission of individual mitochondria. They go together and split apart, sharing contents, forming networks, responding collectively to conditions in the cell. The communication is mitochondria to mitochondria, independent of the nuclear genome. They are something that does not fit cleanly into the category of organelle, which implies a structure that is fully the cells, and does not fit cleanly into the category of symbiont, which implies a separate organism that chose to be there. They are the residue of a relationship so old and so complete that the original terms of it are no longer visible. The two parties became each other over 2 billion years, and what is inside your cells now is not the bacteria and not the host, but the result of that becoming, which is a third thing that has no good name: neurons that have never stopped. Somewhere in the brainstem, a rhythm is going, not a rhythm that started when you woke up, not a rhythm that started when you were born, a rhythm that was established before birth in the neural circuits of the developing brainstem, a rhythm that has been generating the breathing pattern since before the lungs had air in them, running the motor program for respiration in a loop that has repeated approximately 15 times per minute for every minute of your life, for the entirety of your life, without stopping, without a significant break, through sleep and waking and every other state the nervous system has been in. The neurons doing this are in the pre-Butzinger complex, a cluster of cells in the medulla oblongata. They go. They have been going since approximately the 3rd trimester of fetal development. They are going right now at whatever rate the current CO2 level and blood pH and sleep state are dictating, and they will continue to go until the system that supports them fails. The brainstem is the part of the nervous system that does not care about consciousness, not dismissively, literally. The functions of the brainstem, respiration, heart rate modulation, blood pressure regulation, the control of swallowing and vomiting and coughing, the maintenance of arousal states, the coordination of eye movements, run in a state of relative autonomy from the cortex. Damage to the cortex, even severe damage, can leave brainstem functions intact. A person can be deeply unconscious and still breathe, still maintain blood pressure, still show basic brainstem reflexes. The cortex is not driving these systems, it modulates them. The driving is happening elsewhere, in circuits that have their own rhythms and their own internal regulation. The pulse expands. This is true of more than the brainstem. The cerebellum, which coordinates movement, receives input from the cortex but also processes it largely autonomously, refining motor commands on a timescale faster than conscious control could operate. The basal ganglia, which are involved in action selection and habit, do much of their work below the level of conscious awareness. The autonomic nervous system, the sympathetic and parasympathetic divisions that regulate heart rate, digestion, pupil dilation, gland secretion, runs almost entirely outside conscious control, with the cortex having only indirect and limited influence. Most of what the nervous system is doing, most of the time, is not accessible to the person the nervous system belongs to, the neurons themselves. A neuron in the auditory cortex that has been responsive to a particular sound frequency since early childhood has been maintaining its connections, its synaptic weights, its pattern of baseline activity for decades. It goes when the sound arrives. It also goes, at a lower rate, when the sound does not arrive. The baseline firing that keeps the cell alive and maintains its position in the network. Neurons that do not fire go. Synapses that are not used are pruned. The nervous system maintains its wiring through use, and what is not used disappears. This means that every neuron that is still in the adult nervous system has been active, has been going, has been participating in the network's activity for the duration of the person's life. Not constantly, not always at high rates, but continuously enough to stay. The neurons in the hippocampus that formed the memory of a childhood event have been holding that information in the form of synaptic weight changes across a distributed network for the entire interval between the event and now, not actively rehearsing it, not generating a continuous signal about it, holding it in the sense that the physical structure encoding the memory is maintained through the ongoing activity of the cells and the ongoing molecular processes that keep the synaptic proteins in place. Consciousness is somewhere in all of this, not the whole of it, not most of it. The cortical activity associated with conscious experience is a portion of what the nervous system is doing, and it is not the most energetically demanding portion. That distinction arguably goes to the default mode network, which is the set of regions that are most active when the brain is not engaged in a specific task, when the mind is wandering, when the person is apparently doing nothing. The brain is most active when it appears to be resting. The default mode network goes when attention is unfocused. It goes when you're lying in the dark, half asleep. It goes between thoughts, in the spaces the mind fills when it is not directed anywhere. What it is doing in those spaces, the function of the default mode network, is still being worked out. Self-referential thought, prospection, memory consolidation, some combination of all of these. The point is that it is going, and it is costing energy, and it is not quiet just because consciousness is not focused on anything in particular. The nervous system does not rest. The cells rest individually in cycles of activity and relative quiet. The system does not. Something is always going, somewhere in the network, in the same way that something is always being synthesized somewhere in the cell, and something is always being patrolled somewhere in the blood, and the whole body is a collection of continuous processes that do not wait for attention to arrive before proceeding. The breathing rhythm in the brain stem, the baseline firing in the cortex, the default mode network going in the spaces between thoughts, all of it running, right now. In the same body that is, at this moment, doing the optional thing of being aware of any of it. The gut knows things. From far enough away, the gut looks like a tube. That is the simplified version, a long, continuous passage from mouth to anus, specialized in different sections for different stages of digestion, lined with different cell types, producing different enzymes and acids and motility patterns along its length. A processing system. Input at one end, output at the other, transformation in between. The metaphor of the tube is not wrong. It is just the wrong level of detail for what is actually happening inside the wall of the tube. Inside the wall is where the chapter is going, and it takes a while to get there. The enteric nervous system is named for the gut, enteron, Greek for intestine, and it is a network of neurons embedded in the wall of the gastrointestinal tract from the esophagus to the rectum. The estimate of 500 million neurons is the one that tends to appear in the literature, though some estimates go higher. For context, 500 million neurons is roughly 5 times the number of neurons in the spinal cord. The enteric nervous system is not a minor outpost of the brain. It is a substantial neural structure in its own right. It was called the second brain in a 1998 book by Michael Gershon, and the phrase stuck in popular writing, and the narrator is not entirely comfortable with it, because it implies more than is known, and what is known is already odd enough without the framing. What the enteric nervous system does, clearly and without controversy, is manage gut function. It controls the muscular contractions of peristalsis, the wave-like movements that push contents through the digestive tract. It regulates the secretion of digestive enzymes, mucus, and fluids. It coordinates the activity of the intestinal immune system. It responds to the chemical environment inside the gut, the presence of particular nutrients, pathogens, or irritants, and adjusts motility and secretion accordingly. These functions happen independently of the brain. A segment of gut, removed from an animal and kept alive in a dish, will still produce coordinated peristaltic contractions. The control is local, It does not require central input. The vagus nerve connects the gut to the brain, and approximately 80 to 90% of the fibers in the vagus nerve carry information from the gut to the brain, not the other way around. The gut is sending signals up. The brain is mostly listening. The orbit comes around to the word nose, and the narrator is uncertain about it. The gut contains sensory neurons that detect mechanical stretch, chemical composition, temperature, and potentially microbial signals. It contains motor neurons that produce the muscular activity of digestion. It contains interneurons that connect sensory and motor functions and allow local processing and coordination. It produces and responds to neurotransmitters that are also used in the brain: serotonin, dopamine, GABA, acetylcholine. Approximately 90 to 95% of the body's serotonin is in the gut, where it plays a role in regulating intestinal motility and in signaling to the vagus nerve. What does it mean for a neural network to know something? If knowing requires consciousness, the question is unanswerable at present, because we do not have a reliable method for determining whether any neural system other than a human cortex generates conscious experience, and we barely understand it for the cortex. If knowing means processing sensory information and generating appropriate responses, the enteric nervous system does that locally without reference to the brain continuously. The gut-brain axis, the bidirectional communication between the enteric nervous system, the gut microbiome, and the central nervous system, is an active area of research, and the narrator wants to be careful here, because the popular version of this topic has outrun the evidence in some directions. What is established? The vagus nerve carries gut signals to the brain, and those signals influence mood and stress responses and appetite. The gut microbiome produces metabolites that can cross into the bloodstream and reach the brain. Disruption of the microbiome in animal models produces changes in anxiety-like behavior and stress responses, and these effects can be reversed by restoring the microbiome. Human studies show correlations between gut microbiome composition and mental health outcomes, though correlation is not mechanism, and the mechanisms are still being worked out. What is not established? That the gut thinks in the way thinking is usually meant. That gut feelings are the enteric nervous system's opinions about decisions. That probiotics treat depression. The popular version has moved faster than the data, and the narrator finds this uncomfortable, because the actual story, a large autonomous neural network in the gut, bidirectional communication with the brain, microbiome to brain signaling, 90% of vagal fibers going up rather than down, is strange enough without additions. The orbit lands softly on the unresolved part. The gut knows the chemical composition of its contents before you do. It detects the arrival of fats and proteins and sugars and begins adjusting its response: enzyme secretion, motility rate, blood flow, before any signal has reached the brain. It responds to pathogens with inflammation and accelerated transit before the immune system has mounted a formal response. It modulates its own serotonin levels in response to conditions the brain is not yet aware of. The word knows is sitting there, and the narrator is not going to take it back, and is also not going to defend it as precisely the right word, because the question of what knowing requires and whether the enteric nervous system has what is needed is one of those questions that the science is genuinely inside and has not come out the other side of yet. Something is being processed in there. The processing is sophisticated. The results go up the vagus nerve and into the brain. What happens after is what you sometimes call a feeling, and what it felt like before you knew about the vagus nerve is the same as what it feels like now. Apoptosis. A cell is about to go, not because it is sick, not because something external has damaged it, because the signals it has received from neighboring cells, from the tissue environment, from its own internal monitoring systems, have reached a threshold that triggers a program the cell has always carried, a program that exists in every nucleated cell in the body, a molecular sequence that, when initiated, ends in the cell's own dissolution. The program is called apoptosis. The cell is about to run it. It has run in trillions of cells before this moment, in this body, since before birth. The initiation can come from outside or inside. From outside, neighboring cells or immune cells send signals, Fas ligand binding to Fas receptor, tumor necrosis factor binding to its receptor, that activate the apoptotic cascade. From inside, the cell itself detects DNA, damage that cannot be repaired, or a failure in normal cell cycle progression, or stress signals from mitochondria indicating that the cell's own energy supply is compromised. Mitochondria, which in a previous chapter were described as the ancient bacterial residents of the cell, play a central role in apoptosis. They release cytochrome C into the cytoplasm when the cell receives certain internal stress signals, and cytochrome C triggers the assembly of a complex called the apoptosome, which activates the caspase enzymes that do the actual dismantling. The caspases are proteases. They cut structural proteins. They cut the nuclear lamins, the proteins that maintain the shape of the nucleus. They activate enzymes that cut the DNA into regular fragments. The cell begins to compact. The nucleus condenses. The cell membrane starts to bubble, forming structures called blebs. The cell fragments into apoptotic bodies, membrane-wrapped packages of cellular contents that neighboring cells and macrophages will engulf and recycle. The whole process takes somewhere between 1 and 3 hours from initiation to completion. At the end, there is no debris, no inflammatory signal, no local damage to the surrounding tissue. The cell is gone and the space it occupied is clean, already being managed by the cells around it, which have not stopped doing what they do. Apoptosis is a quiet process. Not quiet in the sense of peaceful, the molecular events are rapid and extensive, but quiet in the sense of generating no alarm, producing no distress signal to the surrounding tissue, going without announcement. This is the distinction between apoptosis and necrosis, which is the other main form of cell death. Necrosis happens when cells die from acute injury, lack of oxygen, toxins, physical damage. The cell membrane fails rapidly, the contents spill out, the surrounding tissue responds with inflammation. Necrosis is loud in the biological sense. It recruits the immune system. It produces swelling and pain. It makes itself known. Apoptosis does not. The scale of it is what takes time to hold. The human body loses somewhere between 50 and 70 billion cells to apoptosis every day. Every day. This is not a crisis. It is the normal running of the system. The cells being removed are cells the body no longer needs in their current state, or cells that have accumulated too much damage to function reliably, or cells that are at the end of their normal lifespan. The cells lining the gut are replaced roughly every 4 to 5 days. Skin cells are shed continuously. Red blood cells live for about 120 days and are then removed by macrophages in the spleen. The entire system is in continuous turnover, with apoptosis providing the clean removal mechanism that keeps the cell populations at the sizes and compositions the tissue requires. The cells being replaced are being replaced by new cells produced by stem cells in each tissue. The gut epithelium turns over completely every few days because the intestinal crypts contain stem cells that are continuously producing new epithelial cells that migrate up the villus and are shed at the tip. The body you have now is not the body you had 5 years ago, in the sense that most of the cells are different cells. The exceptions are some neurons in the brain, some cells in the heart, and the lens of the eye, which is made of cells that are not replaced and have been there since before birth. There is a version of apoptosis that happens during development, and it is the version that makes the mechanism most visible in its necessity. The fingers of the human hand are initially webbed. During fetal development, the hand forms as a paddle-like structure, and the spaces between the developing fingers are filled with tissue. The tissue is removed by apoptosis. Cells in the interdigital spaces receive signals that trigger the program, and they go cleanly over a period of days, and the fingers separate. The shape of the hand depends on the death of cells that were built in order to be removed. The architecture of the body in several places is built by subtraction. Apoptosis is not the failure of cells to survive. It is the mechanism by which structure is sculpted from the available material. The hand exists because those cells went. The virome. Something is replicating inside a bacterium in your gut right now. Not a human cell dividing. A bacteriophage, a virus that infects bacteria, that has injected its genetic material into a Bacteroides cell that is using the bacterium's own molecular machinery to copy itself, that will, within 20 minutes or so, have produced several hundred copies of itself, that will then lyse the host cell, burst it, and release those copies into the gut lumen, where they will diffuse and find new bacterial hosts. This is happening in your gut. It is happening continuously. The gut virome, the community of viruses in the gastrointestinal tract, is populated predominantly by bacteriophages, viruses that infect bacteria rather than human cells, and they are doing what bacteriophages do, which is infecting and replicating and killing bacterial cells in a continuous cycle that has been running since the microbiome was established. Zoom out slightly. The gut virome is part of the larger human virome, which includes all the viruses present in and on the human body at any given time, in the gut, on the skin, in the mouth and respiratory tract, in the blood, and integrated into the genome itself. The field of viromics is young. The first comprehensive attempts to characterize the human virome date from the 2000s, made possible by metagenomic sequencing, techniques that sequence all genetic material in a sample without needing to culture individual organisms. What the sequencing found was that the majority of viral sequences in human samples do not match any known virus. The virome is mostly uncharacterized. Most of the viruses in the human body are not known viruses. Zoom out further to the genome. 8% of the human genome is estimated to be of viral origin. These are endogenous retroviruses, ERVs, the integrated remnants of retroviral infections that occurred in the genomes of human ancestors, some of them millions of years ago. Retroviruses integrate a DNA copy of their genome into the host cell's chromosomes as part of their replication cycle. If a retrovirus infects a germline cell, a sperm or egg, The integration is passed on to every cell of every offspring indefinitely, as long as the lineage continues. Most ERVs are defective. The sequences have accumulated mutations over millions of years that prevent them from producing functional viral particles. They sit in the genome as molecular fossils, copied faithfully along with the rest of the DNA in every cell division, doing nothing, or doing nothing obvious. Some are not doing nothing. Synchrotins are a class of genes derived from endogenous retroviral sequences, specifically from the envelope protein gene of ancestral retroviruses. In placental mammals, including humans, syncytins are expressed in the placenta, where they are needed for the fusion of cells that form the syncytiotrophoblast, the layer of the placenta that interfaces directly with the maternal blood supply and mediates the transfer of nutrients and gases between mother and fetus. The placenta in humans depends on a gene of viral origin. The gene was captured from a retrovirus at some point in primate evolutionary history and was repurposed for a function that is now necessary for mammalian reproduction. The same type of viral protein that viruses use to fuse with host cell membranes, enabling viral entry, is used in the placenta to fuse cells that need to become a continuous layer. The biology of viral membrane fusion was taken and redirected. This happened once in primate history, and possibly independently in other mammalian lineages. The viral origin of syncytins was discovered in 2000. It was not expected. Zoom all the way out to what this implies. The human body contains a virome that is largely unmapped, in which most sequences are unknown, which includes bacteriophages actively shaping the microbiome, which includes latent viruses in a persistent but non-replicating state, which includes integrated sequences in the genome that are being expressed as functional human proteins, which includes 8% of the genome itself that is retroviral in origin and is sitting there in every cell, in every tissue, in every moment of every person's life. The boundary of the body, which seemed to shift in the chapter about bacteria, shifts again here. The genome, which feels like the most intimate, most essentially you thing in the body, the thing from which you were built, the thing that makes you you rather than someone else, is 8% virus, not contaminated by virus, not damaged by virus. 8% is virus, integrated, heritable, in some cases functional, in some cases necessary. The virome is not in the body. It is, in part, the body. What the rest of it is doing, the uncharacterized majority, the bacteriophages in the gut, the latent viruses in the mucosa, is still being worked out in a field that is young enough that the basic inventory has not been completed, that is finding new viral sequences in every new sample that is sequenced, that has not yet arrived at any stable picture of what the human virome is, in total, across tissues and individuals and ages and geographic locations. The picture is coming. It will take a while. What it has already shown is enough to change the question. Still running. Something has just been sitting with you for a while, not information exactly. More like the gradual accumulation of a single fact in different forms. The bacteria, the neurons, the mitochondria, the viruses in the genome, the cells dying cleanly while you are unaware of them, the immune system's archive of things you have forgotten, the gut going about its own business on 500 million neurons, while the brain dealt with other things. All of it the same fact approached from different angles. The body is a running process. It does not require attention to run. It has been running since before attention was possible. That is still true right now. The breath going in and out is the brain stem. The warmth in the hands is metabolism. The weight of the body against whatever it is resting on is partly the ongoing result of cells maintaining their structure against the pull toward disorder. The microbiome in the gut is doing what it does. The macrophages in the tissue are patrolling. The memory T cells are waiting in the lymph nodes, still configured for antigens encountered years ago. The body is not waiting for this narration to end before resuming its business. It has been doing its business throughout. There is something about the scale of it that takes a long time to settle. Not the large scale, the whole body, all the systems, the staggering complexity of the whole, the small scale, a single cell, doing its continuous work in the dark inside a body that is itself doing its continuous work, inside a world that is going on regardless. The cell does not know about the body. The body does not know about most of what the cell is doing. The awareness, such as it is, sits at the top of a very tall stack of processes that have nothing to do with being aware. The awareness is optional. The running is not. This is the last chapter, and it is not going to end with anything large. It is going to end with the breath. The one going on right now, managed by the neurons in the pre-Butzinger complex that have been running since before birth, running through sleep and waking and every state the nervous system has been in, running right now, quietly, without requiring anything from the person the nervous system belongs to. In and out, in and out. The same rhythm, the same cells, still going. Everything in this narration was already happening when it started. It is still happening now that it has finished. The ending of the narration does not end anything that was running. The body continues, as it has always continued, as it will continue, in the quiet and the dark and the ongoing fact of being alive, which does not require being noticed, which was never waiting for that, which is simply the thing that is happening right now in you, without pause, without announcement, without end.